Good day and welcome to Lab Talk for this Thursday, May 11th, 2023. We're very excited about this topic today. I think it's going to hit a lot of labs, a lot of people that work in labs and scientists around the globe that work with cell culture. So we're going to talk about the importance of pH in cell culture applications. And we have a treat for you today because we have Ms. Gail Glykoff here. And Ms. Gail Glykoff is our applications lab manager for our water analysis instruments here at Thermo Fisher Scientific. My name is Angela Carnwright. I'll be your moderator today. So without further ado, I'll hand it over to Gail and let her kick us off with this awesome talk.
That's great. Thanks, Angela. So welcome to this lab talk. I'm glad to be here with you today. We're going to start by highlighting some examples of how pH is important for cell culture. Then we'll run a live demo with the help of my colleague, Steve Krynak, who is on site at the Thermo Fisher Scientific Immersive Lab today. We will be using a Thermo Scientific Orion pH meter, an Orion Ross Ultra Triode, and Orion solutions and buffers. So together, Steve and I will show you how to achieve a good calibration and how to get it right the first time. So pH is one of those measurements like temperature and weight that just about every lab makes. pH measurements are so common that we can forget sometimes how important they are. There are 5 examples that we will touch on today of why pH is important in cell culture, including the importance for cell culture media, transfection efficiency, analysis of recombinant proteins, epigenetic analysis, and biological buffers. Reason 1, as we know, biological processes tend to be very sensitive to pH. When culturing cells, metabolic activity generally acidifies the media, and buffering is required to prevent significant pH disturbances. For example, media may be buffered using a non-volatile buffer like HEPEs. The concentration and the titrated pH will impact the buffering capacity. Reason 2, nearly everyone who works in cell culture will also do transfection. However, the effect of pH on lipoplex formation and transfection efficiency may not be widely known. For example, in a 2021 study, the use of alkaline pDNA solution allowed more pDNA bound to cationic liposomes to enter the cells. Reason 3, when working with proteins, pH affects solubility, structure, and stability. Every step of the production has pH-dependent variables. For example, a protein may fold improperly if its isoelectric point and the pH are misaligned. Reason 4, highly complex experimental protocols are used for investigating gene regulation networks. We control pH of reagents to minimize proteolytic degradation. For example, during a chip assay, the wash buffer composition, salt concentration, and pH can impact the binding affinities of antibodies used to detect the protein of interest. Reason 5, given how important pH is for all these processes, the use of buffers can be the key to maintain a desired pH. Some biological buffers have a significant temperature dependence. For example, if the pH of tris buffer is adjusted to 8.3 at 20 degrees C, the pH will be 8.8 at 4 degrees C and 7.7 at 37 degrees C. This is a significant pH deviation of 0.5 units or more. So we'll want to prepare the buffer at the temperature at which we plan to use it. To set ourselves up for success, we need our pH measurements to be quick and accurate. We want a good calibration, and we want to get it right the first time. The best way to achieve that is to follow these 10 best practices. We'll demonstrate the best use of pH calibration buffers, electrode storage solution, temperature correction, storage bottles, filling our electrode, rinsing, blotting, stirring, and maintenance. And now we'll go live to our immersive lab with my colleague Steve Krynak to demonstrate these 10 practices. All right. Hello, Steve. Here we have a complete pH setup, including the necessary solutions, storage bottles, the pH meter, and the electrode, and my lovely assistant, Steve Krynak. So Steve, let's start by examining our pH buffer bottle. So each pH buffer bottle will have an expiration date on it. If the buffer has expired, we should not use it. If it's approaching the expiration date, it's a good idea to order more buffer now. After being opened, it's generally best to use the buffer within a few months for best integrity. We can write an opened on date on the bottle, and that may be helpful. So an important thing to do is to discard any used buffer that we might have in the work area. A fresh pour of buffer is needed each day. We cannot use yesterday's buffer for today's calibration and get best results. So Steve, let's pour some fresh buffer today. So we are going to do calibration at pH 4, 7, and 10. And Steve's going to pour fresh buffer so that we can get our best calibration. After he's poured the buffer, we'll show you the proper way to remove the electrode from the storage sleeve. This will be a tip that's especially good for sealed gel-filled electrodes. So what we're going to do is first we're going to unscrew the storage bottle cap and remove the probe from the bottle. And why are we doing it that way? Now we can slide the cap off the electrode. This is especially good for low maintenance gel-filled electrodes. It will avoid creating a vacuum that can pull the gel out of the electrode when we're coming out of the storage bottle, and avoid pushing unwanted air bubbles into the gel around the junction when we're going back into the storage sleeve. Okay. So Steve has his electrode here. Steve can examine the pH electrode, make sure that the bulb isn't cracked or chipped, that there's no crust or coating, and that it's not discolored. The junction should be present, intact. There shouldn't be any crusting or blockage or discoloration of the junction. And the fill solution chamber should have fill solution in it. Shouldn't be dried out. There should be no crystals, mold, or particles inside. Okay. So I see, Steve, you've opened the fill hole. Can you just demonstrate the fill hole there? Yeah, there you go. So when it was stored, he had that closed, and now he's opened that. And that just allows for the electrolyte fill solution to flow down into and saturate the junction. Great. Okay. Now, just before we calibrate, let's just take a quick look at the electrolyte fill solution and make sure that we have plenty in there. Steve probably already did this, but if you can just show that to the camera, Steve. Yeah. So there's fill solution inside the electrode. He's pointing there. We want to make sure it's filled right up to the top at the bottom of the fill hole. And he would use that Ross filling solution that came with the electrode. So it's important to use the right fill solution. That's great. Thanks, Steve. All right. So he's placed his electrode in the holder, and he's got a stirrer there. We don't need a separate ATC temperature sensor because this electrode that he's using, the Ross 8157 triode, has an integrated temperature sensor. Okay, and the stirrers are going to help us make a faster reading and reach equilibrium quicker. So Steve rinsed the electrode, and now he's got his electrode right into the first buffer. We're going to start the calibration. There we go. So the stirrer will automatically start stirring. The Orion meter will automatically recognize the buffer, read the temperature and the millivolt value of the buffer, and assign the correct pH value for that buffer at that temperature. This is one reason why using a triode or an ATC is important. All right, so it looks like you have the ready signal there, Steve. So he can accept that, and there you go. So he's going to take the electrode out, rinse it with plenty of reagent grade water here. He'll rinse the electrode and the stirrer, everything that extends into the solution, and just blot gently with a little bit of paper towel there, or a wipe. And now we're going on to our second electrode, our second buffer solution. And here we go. So just like the first one, the meter will take a reading, will auto-recognize the pH 7 buffer, read the temperature, and assign the correct value for that buffer at that temperature. And this will give us our most accurate calibration. Here we go. And he's accepted that. And we'll repeat the process. Rinse the electrode and the stirrer, everything that goes into the beaker. This will be our pH 10 buffer. He selects start, waits for the meter to identify the 10 buffer, and go into the ready mode. Once that's done, he'll accept that. And this isn't something we want to rush. We want to give plenty of time so we can have an accurate and stable calibration. And he accepts that, and we are done. So Steve, let's show the calibration results. So the meter will show the average slope. Ideally, that will be between 95 and 101%. If it's lower than that, down to 92 or 102, we might want to perform some maintenance and check the calibration buffers and consider repeating the calibration. If it's not within 92 to 102%, we'll want to replace the electrode. We're also showing here that there are two different slopes displayed, the slope between 4 and 7 and the slope between 7 and 10. Ideally, these percentages will be close, generally, ideally within about 2% of each other. If the 7 to 10 slope is low, the pH 10 buffer may be degraded and may need to be replaced. So Steve, I think we have time for a verification, calibration verification, if we want to move into that. So depending on your quality assurance procedures, you may want to run a calibration verification, or you may be required to do that. So what we can do is we can use a pH like 9.18 buffer, a pH buffer that we have not calibrated with. And we can read that buffer and verify that it reads as expected. Temperature will play a role here. So depending on the temperature being higher or lower, the buffer may read higher or lower than the value on the bottle. So a good way to check this is if there's a chart on the back of the bottle, we can check to see whether or not it reads what we expect at that temperature. In general, we would like our pH to be plus or minus 0.05 pH of the expected value at the measured temperature, or at least plus or minus 0.1 pH. And you should consult your QA protocols to find the particular criteria associated with your work. All right. So Steve, let's just show a little bit of maintenance that can happen at the end of the day that will help us make a quick calibration, an accurate calibration. Great. So he'll be rinsing his electrode carefully to make sure it's clean. We can show them how you would change the fill solution if we want to, although we don't have to do that. There you go. Yep. So he's got a transfer pipette. We can place that pipette into the fill hole and we can draw out the old electrolyte fill solution. And then using the squirt bottle, replace that with new electrolyte fill solution. And we can do that once every week or two. There you go. Once every week or two, and that helps prevent any contamination that might have been caused from samples, prevent evaporation and formation of crystals in the electrode, prevent growth of mold, and generally just keep your electrode performing well. Okay, now he's going to cover the fill hole. Great. And show us how to get the electrode back into the storage bottle without causing any air bubbles. So he'll undo the cap, slide it onto the electrode body. There we go. And then place the electrode back into solution, making sure that the bulb and the junction are immersed in the storage solution. And then put the cap on. Great. Now the storage solution itself is should be changed periodically. So all we need to do is just dump the storage solution out. We can do a quick rinse with some reagent grade water and then refill it. And for similar reasons as the electrode, as we change the electrode storage solution, which is avoid evaporation of the storage solution, avoid contamination. If we've put an electrode in there that wasn't completely clean, we want to avoid it evaporating and making crystals, and we don't want any mold to be growing in there and messing with our electrode. We have mentioned not putting a dirty electrode away, so there's some simple ways to clean our electrode. We can simply rinse it with a 1% laboratory detergent solution, or if we want some more specific cleaning, we do sell a kit that has 1 bottle each of our cleaning solutions. So we have a general acidic cleaner, like pH Cleaning Solution C. That's a good choice. We have a protein contamination cleaner, which is our pH Cleaning Solution A. Then we also have Orion Cleaning Solution B, which will remove bacterial contamination. So you can buy a kit, a pH electrode cleaning kit that has all 4 of these bottles, which is a handy thing to have. All right. Well, thank you, Steve. Appreciate you showing us. Yeah, thanks. Showing us how to do that. So in summary, pH is important for cell culture. The right pH is critical for cell health and maximized procedures. We can correctly calibrate our pH measurement system the right way the first time and follow the best practices for a quick and accurate pH calibration. We have some resources that you can reach out to. We have a perfect pH page. We have some product tours, 3D product tours of Orion VersaStar Pro meter, which is handy. We have a how-to video playlist that shows you how to maintain and clean our electrodes. And we have a great pH measurement handbook.